genes in development. So we're gonna breeze by the gene stuff a little bit because I don't think it's as important as talking about central nervous system development. So that's what we're gonna focus on. Okay, so genes. Many of you have had at least one class, either in college or high school, that talks about genes. So we are going to, like I said, sort of breeze by this sort of introductory information as far as central nervous system development, because I want to make sure that I have enough time to go through the development stuff. And perhaps Dr. Kaiser goes through some of the other stuff related to that. Okay. Okay. So you have genes. You have 46 of them. You need a full dresser for all 46 pairs. That one's free. Okay. So genes control everything about us. Okay. They control what we look like. They control our development. They control how a lot of how we think, a lot of what we do. They control a lot of stuff. Okay. We're going to quickly show the example of facial expressions and how our facial expressions are basically hard-coded into our genes, which tells our face muscles how to act in various cases. Now, those could be those could have deficits, but it's not common, those deficits, right? So here are facial expressions, okay? And these faces are of people with deficits, okay? These faces are of people with deficits. And I realized that I need to get better pictures. These are not great images. They're very pixelated. But these are facial expressions of people with deficits, and you can still sort of tell what expressions they might be making in each of these cases. And the deficits still show evidence of facial expressions because they're trying to make the facial expressions, even though they might not look like something that you can produce. Okay. One fundamental thing that I want to point out with genes is that they don't directly produce behaviors, because that also requires things like nurture. Okay, things like your environment. Okay, so genes, their only deal is to produce proteins. That's it. That's all they do. The accumulation of proteins in your body lead to development, make things grow, that sort of thing, okay? And so when we talk about genes and psychology, we're essentially talking about indirect effects, okay? And let's not forget the and here, the interaction with the environment. Okay. Interaction with the environment. Don't forget that. And that's going to affect the proteins, the genes that are turned on or off through epigenetic factors. But it's also going to be the reason for the season on the way genes are expressed. Okay. So I've talked about evolution a lot in the last week. I'm kind of evolutioned out. So the idea here is that evolution is a process in which organisms change over time. And the change either impacts positively or impacts negatively. If you get to procreate, if you get to replicate, if you don't. So some common misconceptions that I want to point out. Lamarckian evolution is completely wrong. The use or disuse of a structure or behavior causes an increase or a decrease in that behavior. A 9, incorrect. No. A change at a single generation does not necessarily mean that change will be carried over to offspring, the next generation. Jean-Baptiste Lamarck, sorry. Come a little bit wrong on that one. Humans have stopped evolving. Well, unfortunately, the time scale for evolution is geologic. So we're talking billions of years. And for humans and human ancestry, we were talking about, well, it depends on how far you go back. It could be millions of years, but it's definitely hundreds of thousands of years. Humans have not stopped evolving. We just can't detect it with this slow of time scale, with how slowly we move through time. Okay. Evolution does not mean improvement. Now, evolution tries to allow an organism to be more adaptable to environmental selective pressures, but generally speaking, that's not really the case. Okay. Evolution acts to benefit the individual or the species. Again, I mean, these are two sides of similar coins. Okay. There have been many instances where evolution has not benefited a species. So it's likely that if not for the big asteroid that hit Earth and decimated the dinosaur population, it's likely that if that didn't happen, dinosaurs probably would have eventually died out. But because it did, their large size and their need to consume lots of food, not so great. So there have been 5 mass extinction events in the history of the Earth. And we're talking about extinction of life. It's entirely possible we are in the midst of the 6th mass extinction. And that's partly due to humans. So cool. The mass extinction event, by the way, is specifically about mammals and specifically about humans. So, we're doing great. Okay. All right. So we are going to take this broad idea of evolutionary psychology / evolutionary theory to explain brain and central nervous evolution. Okay. Central nervous system. Yeah, we're in the middle of the 6th. Yep. So if you think about it, and I talked about this yesterday with the handful of you who are in history of psych. So there's lots of differences in humans, right? We are a varied species. Quite significantly so. I mean, consider dogs and consider cats. They're also fairly varied. And I'm not even talking about big cats, just house cats. Incredibly varied, right? Dogs, not wolves, incredibly varied, right? So all of that stuff has to do with how we've developed through time, not how humans develop from zygote to an adult and an older adult and all of that stuff. Not just that, but like, where have we come from? What have our ancestors done that lead to things that we do today, right? So things like the differences in peripheral or color vision, what our sleep mechanisms are, what they're for, and why they do the things that they do. Eating habits and temperature regulation, things like all of that, even mating, even reproduction, Oh, I just said mating. Thinking about things, problem solving, heuristics. I talk about those a lot if you've been in a class of mine. So let's take all of that, crumple it down, and just specifically talk about how the brain has evolved and develops. Okay. So there have been a good number of things that humans have done, including what human ancestors done. So our shared ancestry with other primates that led to the development and expansion of the brain. Now, it is one explanation. That's why it's underlined. So it's not the only explanation. But specifically, what I wanted to point out is that brain development and the size of our brain requires 20% on any given day of resources. So sugar, oxygen, as I mentioned a couple of weeks ago. And things that we did to increase the size of it was learning how to use fire to make food easier to digest. So to cook food, to break down proteins, because that's what cooking does. It denatures proteins, it breaks down DNA, and that makes all of it the easier to digest. It makes it easier for hydrochloric acid in our stomach to break apart those denatured proteins, because that's what enzymes and acids do. They break down molecules. And so when you cook it, you come in with already denatured proteins, you put it in, it allows you to digest it easier. Great. Doing stuff in groups. So becoming kin, banding together. In addition to hunting in groups versus hunting in solo, right? It allows us to come around and attack and do different maneuvers and all of that kind of thing. And yes, the development of tools, of course. And then through all of that, our body was also figuring out ways to increase glucose transport. So there you go. All right. So let's start from the very beginning. One of the first structures that develops in the zygote is the brain. So it starts off as this neural plate. Let me grab my mouse here. This neural plate here. It kind of looks like a chest and abdomen, doesn't it? Like this is a chest and this is a six-pack. You know? It's kind of funny. So it starts out with this brain plate, and then what it does is it folds into itself and becomes the neural fold, okay? So it becomes a tube. So this edge here and this edge here are folding inward, right? So in the depth plane, so if that's the neural plate, it folds inward, okay? And so it becomes the neural tube with the neural groove, the neural fold, and the neural plate on the inside. So this is going to be both the brain and the spinal cord. And it's going to start differentiating as time progresses. So the neural plate begins to open just a little bit, sort of expands like a flower blooming almost. Okay. And then it's going to stretch out. You've got your heart bulge that comes off of that. Okay. You've got this neural tube that's going to become your spinal cord. Hooray! And then let's expand out the time scale here. So at 3 weeks, the zygote now has a set of neurons that have begun begun differentiating into hindbrain, so your pons, medulla, your midbrain, okay, the tectum, the tegmentum, etc. Okay, the colliculi, all of that. And then the forebrain begins to advance. And that the forebrain includes your limbic system. So we've got the basal ganglia and the caudate, thalamus, hypothalamus, as well as the cortex. Okay, so that's all in this tiny little bulge. Oops, sorry, right here. Tiny little bulge in the front. Just 4 weeks later, those structures are even bigger. Okay, you can even see a little hand and arm. Oh, look at that. I'm sure those of you who are like steeped within development have seen pictures of these constantly. And so here we go. We've got the spinal cord forming. We've got cranial nerves heading toward the face, the face, eyes, and neck. Okay. And you can see here that we've got the pons developing. This is the pons. And down here is the medulla. Okay. And then we've got the midbrain here. Again, this is going to also differentiate into the cerebellum. Okay. Oh, sorry. This is the cerebellum right here. Midbrain gets hidden. And then we've got the forebrain again, advancing, convoluting. It first starts off really smooth. See, there's no convolutions at 11 weeks. Okay. So we've got the forebrain starting to turn into shape. You can see this little divot here. That's going to be your temporal lobe. That little divot there. Midbrain, again, things like the tectum, tegmentum are going to get covered by the occipital lobe that's developing here. It's going to get covered and sort of smooth brain. Yeah. And then we've got our pons, our cranial nerves, pons right here, medulla right here. Okay. And then at birth, this is a fully convoluted. So so 38 weeks is a full term. Okay. So for 27 weeks, what happens with the fetus is the forebrain continues to expand and start folding in on itself, start making all of those convolutions. Cerebellum begins to do the Purkinje cell expansion with all of those. I showed those a couple of weeks ago. They have like all of those dendrites to like, I'm a happy tree, right? So for 27 weeks, the brain just gets more and more folds in it, and it gets bigger and bigger and bigger for 27 weeks. Okay. Now, because part of this class is to do with neurons, while this is happening on the cellular level, damn it. There we go. You have what's called proliferation. So, proliferation begins almost immediately as the zygote and neural tube, the plate and neural tube begin to form. Okay. Some cells will become stem cells, and they will continue to divide undifferentiated. Also, early in development here, during proliferation, you get the creation of your ventricles, which is especially important for the transport of nutrients and the cerebrospinal fluid and things like that. Migration. So not stem cells, but other cells that are being created start moving. They start moving, okay? And this also includes the glial cells that we talked about as well. They start basically moving to where they're supposed to be. They haven't really differentiated into the various kinds of cells yet. That happens during differentiation. Okay. That happens during differentiation. Axons form, dendrites form. Okay. They go when they reach their target, and that's that. And I'll show you how that works with evidence from reptile brains. Okay. After cells have been differentiated for where they're supposed to be, whether they're going to be in the midbrain, hindbrain, or forebrain, those oligodendrocytes, and Swan cells begin their myelination process. So they take fat from the diet of the mother coming in through the placenta and umbilical cord, and they start making those myelinated sheaths. Okay. And this is important because now we're in that 27-week span where the brain is just expanding. So you need transmission to start speeding up. So first and foremost, during proliferation, migration, and differentiation, you have that slow transmission of the action potential. So these are slower transmissions. They're not necessarily needed for anything too heavy. Okay. The last thing is synaptogenesis. So once the neurons have migrated, differentiated, and been myelinated, now they can start connecting to other cells. And that's synaptogenesis. So this is the final progression. Now, you do synaptogenesis all the time. Okay. The vast majority of synaptogenesis occurs in vitro, which is kind of cool, but synaptogenesis happens throughout your lifespan as you learn things, as you get older. Okay. This happens all the time. Okay. As you age, part of the disintegration of your telomeres. So that's according to a lot of geneticists, the decay of your telomeres that hold on to the ends of your chromosomes, once they start to disintegrate and allow for the ends of your chromosomes to unravel, that then gets represented as aging in this process. So part of that process is that synapses don't, new synapses, I should say, new connections don't form as readily. Okay. So the question is, do we get all of our neurons at birth or during in vitro development? Do we get all of them and then it's just like get hit in the head, drink some alcohol, take other drugs, everything? It starts from 100 and then slowly takes it down. That was the early thought, that you had all of your neurons once you were born, but that is incorrect. So we have stem cell evidence, which is the last few decades that stem cells were discovered. So they are undifferentiated neuronal cells. Undifferentiated. They will likely be converted by replication into what are called daughter cells. And those will either be glia or neurons. Okay. Olfactory cells, remember the one cranial nerve that does not go to the back of the brain. They go directly into the cortex. No thalamus for the olfactory. Those constantly get replaced. And one hypothesis regarding that is when you smell stuff, these are literal chemicals that are going into your nose. So one of my favorite things to say, which really may upset some people, but when you smell poop or farts, I'm sorry, but those are little literal poo particles going up into your nose. You're welcome. That one is also free. I'm 12. I don't know if you recognize that. So when those particles go up into your nose, if they are damaging, so if let's say there's some sort of toxic smell like ammonia or something like that, those damage those neurons. Okay, because they are a toxic chemical, which is why you shouldn't like be in a closed room working with bleach, right? Because if you inhale all of that bleach, it's going to start like not only going into your lungs, but it's also going to be damaging your cells. It's also part of the reason why when COVID takes away your taste and your smell, when COVID takes away, those also regenerate because your olfactory cells regenerate and your taste buds will regenerate. So good news on that front. Okay. All right. So I mentioned that proliferation occurs. And so we need to discuss how proliferation happens. And we're going to use evidence from studies that used reptile differentiation and proliferation. Okay. We're talking a long time. I mean, it's gradual, but because proliferation does have to travel, the tiny little neuron has to go pretty far, depending on where it is and where it's supposed to be. Since the fetus is getting bigger and bigger and bigger, neurons are staying the same size. It's like, crap. I got to go all the way to the prefrontal cortex. Oh, this is going to take forever. And it kind of does. But in 1954, Sperry used some newts. Okay, maybe I guess they're not reptiles. They're amphibians. Are newts? I don't remember. Are newts reptiles or amphibians? I don't remember. But Sperry did some research on newts following the chemicals that are released to basically tell the neuron, keep following this chemical release, and eventually you'll get there. Tor, is this why it takes a different amount of time for everyone? Because some people get it back soon, but I had COVID in November and still haven't regained some. Yeah, so it's going to be dependent on your development. And also, I'm sorry you had COVID. That sucks. I'm glad you're still with us. Amphibians. Thanks, Jessica. Yeah, it does take a long, it can take a long time. Some people, it's really quick. Depends on how severe the damage was. It's also pretty sucky that it does damage. But that's exactly what the hypothesis is that once you damage those cells, it's not like you use your sense of smell forever because those neurons will get replaced eventually. So that's cool. Now, if the mechanism for regrowing olfactory neurons or taste buds is damaged, then yeah, you will lose those senses forever. Or you won't be able to smell or things like that. So we talked about how concentration gradients allow for the inflow and outflow of sodium and potassium in the neuron cells. Well, we're going to use the same principle for this target path. So the chemical path is such that on one side, the neurons is attracted, and on the other side, it's repelled. So we get the movement across. Exactly like, you can think of it exactly like the north and south poles of a magnet, right? So north is attracted to south, but north is repelled by other north, right? So you can think of that kind of attraction with respect to chemical gradients. Okay, so let's take a look at these newts. She turned me into a newt. got better. All right, so here we go. We have the newt. Okay. We have two structures that we're going to talk about. We have the retina and we have the tectum. Okay. And we have our location words. Okay. Everybody remember those location words? Dorsal is the top, ventral is the bottom, anterior is the front, posterior is the back. This is why I use those even with reference to animals. Because normally what you would see in like a heavily neuroscience textbook is rostral and caudal instead, because that's the animal. Okay, because their eye is faced toward their nostril. Look at his nostril. So here we have 4 paths. We have the red path where the anterior portion of the retina, the front part of the retina, goes to the posterior part of the tectum. The dorsal part goes to the ventral part. Ventral goes to dorsal and posterior to anterior. Let's say we cut the optic nerves, which is the bundle of these fibers here, right? We cut that optic nerve, right? And so we take the retina and we rotate it 180 degrees on the vertical axis, okay? So the front becomes the back. The back becomes the front. The top becomes the bottom. And the bottom becomes the top, okay? Everybody see? 180 degrees on the vertical axis. I have now rotated it. And it's been rotated counterclockwise. That doesn't really matter because it's 180 degrees. Okay, so you can track where the blue connection is, where the I guess teal connection is, the red, right, and the orange or yellow, whatever you guys see, whatever color you see. I haven't seen the answer yet on what this wiring diagram is showing. It starts with a C. Okay. Now, within these newts, look at these newts, these awesome creatures. Because of the chemicals, gradients that attract where the connection is supposed to go, look at this. The eye is upside down. The eye is upside down. Now, the eye is going to connect to the same exact places it used to be connected because of those chemical gradients. I think that's crazy. Don't try this at home. Do not cut your optic nerve because yours won't grow back. Newts are far more simple creatures and their stuff will grow back. Okay, but you can see here that the old dorsal connection still goes to the ventral portion. And this, yes, Marissa, contralateral. Very good. The posterior will still go to the anterior. The old ventral still goes to the dorsal portion of the tectum and then anterior back to posterior. They still go toward the connections that they had previously made because of those those chemical gradients. Now, what happens to the newt's vision? It's upside down. Now, that kind of sucks for the newt. One eye is right side up and the other one is upside down. And that's because the vision here is going to be reversed. So what their brain is going to have to do, and I don't know what a newt sees, but what their brain is going to have to do is take a right side image and take a upside down image and sort of put them together. Right? So they're going to have to figure out how this combines. Okay. Couple of things. Actually, yeah. I want to talk about nerve growth factor. So nerve growth factor is a protein. Two folks, Levi and Montalcini, or Cini, Cini, I think it is, Montalcini. These two gentlemen determined nerve growth factor. It is the protein that promotes growth. Okay. Now, if you overproduce neurons, yes, Marissa, that is correct about the contralateral wiring. Yep, they go to opposite sides. Exactly. So if you produce too many neurons, and sometimes you do, there are limited resources, and so sometimes some neurons, it's just too much. Your brain has an optimal size, and if it's too big, that's a problem. Okay, because there's not enough space inside your cranium, which doesn't move. Okay, it does get bigger because your brain, your skull does get bigger as you grow, but it's limited once you become an adult, right? Once you're done with puberty and everything like that. So apoptosis is the way that cells kill themselves. Okay. Apoptosis, one of my favorite biology words, apoptosis. Because they basically explode. Apoptosis. I'm willing to bet that, I know this probably has some Greek roots to it, but I'm willing to bet scientists, biologists who are like, let's name this. They're like, the cell pops. It has nothing to do with that, but I like to think so. Okay. So nerve growth factor gets input from other chemicals. Okay. And one aspect of CNS development is that a healthy nervous system contains no neurons that failed to make appropriate connections, because those neurons are murdered. Okay. So if they are not exposed to neurotropins, which are a kind of neurotransmitter, then they will kill themselves. Okay. So your healthy nervous system contains no cells that can't talk to another cell. Okay. Now, neuronal death is sad. Most pruning occurs during actual development. So up until the age of about 25 is when your brain is fully matured. Once that occurs, apoptotic mechanisms do slow down. Now, you still do lose brain cells. The things that I mentioned earlier, like hitting your head and making your brain hurt. Drinking alcohol specifically, but also things like turpentine. You don't want to drink those either. Other things like other drugs, other psychoactive drugs may destroy neurons. Okay. Now, the coolest thing, cool as I mean interesting, is that people who are congenitally blind, that is, there is no connection between the eyes and the visual cortex, so congenitally blind, they were born that way, have a lot of neurons in their visual cortex. It's thicker. It's filled with neurons. Because ineffective neurons do not get pruned based on learning like non-visually impaired people do. Okay, so if something's not working as far as vision is concerned, we get rid of it as non-visually impaired people. But congenitally blind individuals can't do that because they're not getting feedback from the environment. On the dendrite side, we talked about how dendrites, when they are making new connections, will increase their spining. So this is dendritic expansion, right? And so there are, if you look here, A came before B. So over time, you can see this particular cell, and those are dendrites. Those are dendrites. You can see the amount of branching has increased from A to B. And same thing with this person. You can see that this is 50 microns across, right? You can see these dendrites just even in a month's time. If you are learning and you are alive, that you will increase your amount of dendrite branching. Okay. And just overall, you can increase dendrites just by living your life, but there are other ways that you can increase them too. So there is some evidence that exercise increases spines, so dendritic branching. Okay. Doing cognitive exercises, so reading, crossword puzzles, memory games are also another way to increase dendritic branching. Okay. Now, if a neuron does go through apoptosis for whatever reason, a new one will likely take its place in a normal functioning brain, in a neurotypical brain. Okay. And with the information that is nearby, that is the other neurons, there will be better learning involved because those connections are sort of pre-programmed for that new neuron that took the dead one's place. Okay. Yeah. Don't say muscle memory. The brain cannot be exercised. Yeah, I hate that. Yeah, that's it.